I think, so it's my pleasure today to get to introduce Dr. Scott Latender. I really think he does not need any introduction in the neuro-HIV space. Scott, you're everywhere. You're involved in just about everything, and have been a thought leader in the field. But he's currently at UCSD, and I trying to think of, I don't really, he graduated from Georgetown School of Medicine, and then did his residency in internal medicine at the Naval Medical Center in San Diego. He also completed fellowships in infectious disease medicine at Duke University and in neurologic HIV research at UCSD. He performs translational patient-oriented research of the CNS complications of chronic infections, including HIV. And he really, works as part of a multidisciplinary team, doing everything from large-scale studies to treatment trials, both in the US and internationally. So with that, Scott, I'm going to hand it to you. I really look forward to hearing you speak today.
Domo arigato. So, hi everyone. I was asked in December to do a talk on clinical trials, and I thought, oh, I like that, and so I've kept sort of refining it since then. And Leah and Caroline were looking for volunteers, so I thought, why not? This is not something that we talk about too much, but a lot of opportunities here. So disclosures, and I've shown this slide a lot, but just thought I'd use it as a jumping off point here. I put this together just to remind us of the complexity of the factors that affect the brain and people with HIV, either in categorizing them into viral, host, drug-related, and other pathogen-related factors, but also an element of time that we don't always remember to factor into our thinking about this. And so really the effects start before people are infected with HIV, and has to do in part with social and structural factors. I don't know if I can say that anymore, or but also genetics and things that put them at risk for HIV, like drug use and sexually transmitted infections and so on. After infection, as you know, and what we used to see back in the bad old days more so, was this uncontrolled HIV replication with either no treatment or inadequate treatment, and the immune suppression that goes with that. And there might have been some enrichment for people who were using more drugs or had more sexually transmitted infections, putting them at greater risk for getting HIV. But we're doing a much better job at linking people to care now. And so we diagnose them sooner before, generally, but not always, before their immune system is so injured, and get them on even more effective, less toxic therapy than we used to have. And so we can suppress HIV replication below detectable levels. But there continues to be transcription and translation, a point that I'll bring up again. If they did progress to a low CD4 count, even with immune recovery, they still can have an impact of past immune suppression. And these other factors don't go away, but we can treat sexually transmitted infections, including viral hepatitis. And then there's a long period of clinical stability. I see the urgent walk-in clinic in our Owen clinic here in San Diego, our clinic for people with HIV. And so a lot of what I see week to week are not HIV related, other things that people come in with in this period of stability. But we know that people with HIV are at greater risk for aging related complications and getting them at a younger age. So as they get older, this premature aging process, biological aging, we think 5 to 10 years takes place, and they start to get metabolic syndrome, cardiovascular disease, and brain health disorders as well. So I'm going to show you a few slides about the influence of HIV in this process, even during effective therapy. And Jules and I have had discussions about this over time, and it's a great example of when I see data and I get a little overly persuaded, like by people who think that HIV is no longer a part of the equation. And Jules has, I don't know why I say that in a surprise way, but Jules has said, Scott, no, HIV still is part of this. And he convinced me, and data like what I'm going to show you has convinced me as well. So these are data from Zhang Zhang, as well as Christina Gavignano and Vince Marconi and our old friend Bert Anderson, using the IPDA assay, integrated proviral DNA assay, and comparing CSF-derived HIV DNA to blood-derived HIV DNA, and somewhat surprisingly, but perhaps not so, showing that DNA levels, integrated DNA levels in CSF cells are higher than in blood-derived cells, and pretty much consistently so, but not always. And maybe this isn't surprising because activated cells tend to have more HIV DNA, and they're the ones that are more likely to migrate across anatomic barriers into CSF. And so CSF, if you will, is enriched for activated high HIV DNA cells, whereas those cells are more diluted in the larger population of blood cells. So, but regardless of the explanation, they nicely showed that integrated HIV DNA, proviral DNA levels are higher in CSF cells, and they correlated this with some brain health indicators like executive function and learning. In addition, Sarah Janella and Guancho Zhang at the University of North Carolina generated some beautiful data that was published in 2023 using brain tissue collected by the Last Gift program with some support from the NNTC as well. And as you may know, the important factor, one of the many important factors, but one important one about the Last Gift program is that they do rapid autopsies and they collect tissue within hours of the person dying, which enables some of the assessments like this. And what the group at UNC did, working with Sarah Janella and the Last Gift program, was they were able to separate and isolate different cell populations, including primary brain microglia. And these different symbols in the graphs here are different donors. You can see from cortex or basal ganglia, and showing the amount of total DNA, integrated HIV DNA, and HIV RNA. And this addresses the issue about how well does what we see in CSF reflect what's present in brain. And this doesn't address that directly, but the question is always there, is what we see in CSF, does that really reflect what's going on in brain? And these data at least support that even with a rapid autopsy and very good methods, that scientists do see infected cells in brain tissue. And then they also expose these cells to latency reversing agents, and we're able to show that the amount of HIV RNA in these cells in culture increased when exposed to latency reversal agents. So there's a virus there, and it's inducible. So based on data like this and pharmacology data that we and others have generated, we performed this clinical trial with the idea that the brain health disorders that occur in people with HIV was related to persistent low-level or intermittent low-level HIV replication in the nervous system. And so in the ACTG, we randomized about 200 people who were already on suppressive therapy with that idea that the suppressive therapy was not suppressing replication in the central nervous system. So we randomized them to either placebo, dolutegravir plus Maraviroc placebo, or dolutegravir plus Maraviroc. And the bottom line here, I don't want to dwell on this a lot, but it was a negative clinical trial. We didn't see any difference between the arms in cognitive performance over 96 weeks or in depressive symptoms. So I was, as I tend to do, I was discouraged by this, but Joe Aron, in the ACG said, no, this is great. This shows that this is not due to HIV replication, because if it was, you would have seen an effect. So that's the point you should emphasize, and we did. And so, coming back to this idea that it's transcription stupid, that it's not replication, but we know that in infected cells that are metabolically active, they're going to keep transcribing DNA, right? And maybe I'm the last person to come to this realization, but it's not replication of intact viruses. It's transcription of HIV DNA into HIV RNA and translation of HIV RNA into HIV proteins like TAT. And so there's data there are data that have come out in the last few years. These from Kazuo Suzuki, working with Bruce Brew in Australia, showing similar data to the Gabignano lab that HIV RNA and DNA are higher in CSF than in blood, and that they did magnetic resonance spectroscopy showing that these correlated or were associated with findings on brain imaging as well. And then Avi Nath, all of us know, of course, has been working with TAT and for nearly the entirety of his career. And they developed an assay to measure it in CSF, and we donated some specimens along with other groups, including Avi's own group. And they found that they were able to measure it. It was present in about 37% of CSF specimens. And again, they correlated it with different cognitive abilities, including here the motor functioning and information processing speed. So one last point I want to make just about the virus is it's not just the higher end of the spectrum that can influence people's brain health outcomes. It might be the lower end as well. And so this is just summarizing a couple of data from a couple of projects here in San Diego. This graph, this histogram on the upper left, shows the distribution of HIV RNA levels in people who are not on antiretroviral therapy in CSF in blue and blood in red. And in general, as we know, CSF levels are generally lower, about 1.5 logs lower overall. But there are some people who have more virus in CSF than in blood, about 4%. And importantly, there's a group of people who control HIV RNA and CSF when they have detectable levels in blood. So at one time point, at the first time point in this analysis, it was about 23%, but Mattia Tronfio, one of our junior investigators here who came from Torino, said, Scott, I think it would be a good idea if we just looked at this over time and look at what percent of people durably suppress HIV RNA and CSF, and it's a lower percent, but still 7.5%. So I likened this to sort of people who are elite controllers a bit in blood, that their own immune system can control HIV RNA, and there might be something like that going on here as well, that there is a group of people who suppress HIV RNA in CSF without therapy, and they may have a better antiviral immune response, but perhaps there's also bystander injury from that more robust antiviral immune response. So this is a nice graphic that Mattia made looking at the summarizing correlates of the HIV RNA levels in CSF, including the CSF to serum albumin ratio. But I don't want to dwell on that because we need to move on. But I also want to say that this recapitulates somewhat data from Paula Desplatz with brain tissue specimens from the NNTC, in which she took this brain tissue from people at autopsy, and she measured HIV DNA, HIV RNA, and P24, and found that there was this group of people with very high levels of DNA, RNA, and proteins, but there was also this group of controllers at death who not only didn't have a protein or RNA, but also didn't even have detectable DNA. And there's always sampling issues to consider here. What piece of the brain did she get? Was she just unlucky? But and where she sampled, but it reinforces that there is a group of people who seem to control HIV in the brain. And so we're lumping all these people together, right, into clinical trials because we don't have a good way of distinguishing them. And that's one of potentially many reasons why we've seen failure of clinical trials, or at least negative results. The clinical trials have succeeded, the results were not positive, right? So negative clinical trial results for nearly everything we've looked at. There's some clues here. Our friend Ned Sachter years ago showed that paroxetine improved global performance. There was some indication, I think Giovanni Scafito did this work years ago on Depranil. There's some evidence from switching from efavirenz, but that's probably related to neurotoxicity. And then we shouldn't forget that the world does not revolve only around drugs, that there are also non-pharmacologic interventions, and things like exercise and cognitive behavioral therapy may be beneficial, but not everyone can exercise, and cognitive behavioral therapy doesn't always have durable effects. So we shouldn't forget these, but I'm just going to put those aside for now because it's not the focus for the rest of the talk. So another thing, Leah and I were quipping earlier about seeing each other so much because we have a new project on brain biotypes called MyAD, and we've been having a lot of meetings for that, and that's good. But among the many things it is doing is it's focusing on the reality that the way the brain manifests injury is not limited to cognition. Of course, there are mood disorders, cerebrovascular disorders. We, of course, know there's symptomatic CSF escape. There's all these other disorders, right, that we sometimes blissfully ignore in our analyses, but that can really influence the outcomes and may be another factor in why we've had so many negative clinical trials. So this is a course approach to doing clustering analysis compared to what Leah and Raha and others are doing. But it's a first effort, and it's pretty good. I don't want to diminish it too much, but I just want to admire and respect all the work that Rob Paul and Leah and Raha are doing. But in this, we learned from them, and we said, okay, well, let's start out. Instead of just taking these 7 cognitive domains, let's add in 10 more features of depressive symptoms, cognitive symptoms, and activities of daily living, and apply clustering methods to that larger group of data and see what the clusters look like. And our analyst, Bin Tang, working with Raha Dascape, found 4 clusters as the final solution to this group of data. And about half the people, they were healthy in all 17 of these dimensions. And in group 2, phenotype 2 here, you can see the ones that are elevated here are the depressive symptoms. So there's some differences with the healthy group in these other dimensions, but primarily these are depressed people. If you jump over here to group 4, these people also did well in all of these dimensions, including activities of daily living, but they were very cognitively impaired. So cognitively impaired, but not very symptomatic, not having problems with activities of daily living, which is sort of interesting. And then in this phenotype 3, these people were broadly unhealthy in all 17 of these dimensions. So in a clinical trial of cognitive impairment, we would enroll 3 and 4 probably, and maybe even 2, but there's reason to think that these groups could be very different. And so are we diluting the signal there by enrolling all these people into a clinical trial when the intervention might only work in one of these groups? So there's a nice forest plot here looking at some of the correlates of these unhealthy clusters compared to the healthy cluster, but I like the forest plot, but I'm not going to take you through that because it's sort of not the point today. We've also, with the help of CFAR funds here, purchased an Olink proteomics instrument. And, you know, it's proteomics, but it can only measure 96 proteins at a time. So still advancement from what we previously have had. But you can measure a large number if you do enough panels and have enough money. These data just show the results of the target 96 inflammation panel, and in CSF from about 400 people. And when we compare these same neurobehavioral phenotypes that I showed you on the prior slide, MVP 1, 2, 3, 4, what you can see is this broadly unhealthy group in the heat map on the left, all the red, that differs most from the broadly healthy group, MVP1, the leftmost column. And if you jump over to the columns on the right and you look at global impairment, recall impairment, so memory, and depression, there's differences, but not as dramatic as this broadly unhealthy one. So when we're thinking about inflammation, something that might, treatment that might treat inflammation, if we're using global impairment, the differences with the healthy group are much more modest than this broadly unhealthy group. So again, who we enroll in these clinical trials could influence the results, of course. And on the right are just some of the proteins that differed the most. So again, I don't want to dwell on the specifics today because I want to get on to the more clinical trials. Ron Ellis has also been working with Ben and Raha to analyze longitudinal data over 18 months. And you can see we find, somewhat similar, but somewhat different as well, phenotypes, neurobehavioral phenotypes longitudinally, a stably healthy group rather over about a half, depressed, cognitively impaired group that's improving over time, a stably depressed group with impaired daily functioning, and a worsening cognitive impairment over time. And Ned Sachter tried to sort of address this idea of enrolling people who were changing with the idea that if their cognitive impairment was worsening, then they might be more reversible. They might not have as much irreversible legacy effect. And so this is just to show you that this sort of work is continuing, and we're working toward trying to identify clusters that may be more biologically driven so that we can design a clinical trial to address them. So finally, the clinical trials part of it here, I've categorized, I went through and searched clinicaltrials.gov, reached out to several colleagues by email who were kind enough to not ignore my emails, and put together this list of clinical trials. And the subsequent slides are going to be just sort of going through one or two slides at a time, a bunch of clinical trials, but the point is to make you aware of them and to stimulate thinking about them, and to make the point that we can learn from all of these, whether they're end up being negative trials or positive trials. If we're using sort of a core assessment battery, Leah and the Hopkins group have been working a lot on data harmonization. We can harmonize those data and learn about, maybe learn better about about what is influencing brain health, even if an individual trial ends up having negative results. So again, I've categorized these as trials that have brain health as a primary aim, brain health as a secondary aim, and a focus on treatable traits, which is something that we've been focusing on in a navigating neuro-HIV working group that NIMH organized with Vasu Rao, and we had physicians who work in neuro-HIV from around the world contributing to it. So, and I put this tree on the right here because it's a favorite graphic of mine. I've probably shown it here before, but it's just reminding us that, this idea of, sort of clipping one or two roots at a time or one or two branches at a time may not really solve the problem. And so we may need either combination therapy, sort of, again, focusing on multiple treatable traits, but ideally, if we can identify a central nexus, a central treatment point, perhaps suppressing HIV transcription, right? Then we would fell the tree, that all of these other factors would ideally follow along in a domino-like fashion and improve. All right. So clinical trials with brain health as the primary aim. So I mentioned this first, a little self-serving, but we worked with Rick Meeker and Monica Diaz, along with Ron Ellis at UCSD, on this clinical trial that we got funding for, and Rick and Monica are still working to get final FDA approval to start enrolling. But it's a clinical trial of LM11A31, rolls right off the tongue. But it's a ligand for P75 neurotrophin receptor. And if you look at the pretty graphic on the right that Rick provided, you can see here that this LM11A31 both facilitates beneficial neurotrophin signaling via these pathways, but also inhibits neurodegenerative signaling here as well. And they have nice data from in vitro experiments that Rick does beautifully, as well as animal safety profiles. And they've been looking at it in Alzheimer's, and we're hopeful that once we get permission to start enrolling, that this could be really a valuable and novel intervention. So that's all I'm going to say about that one. I also want to really recognize all the fantastic work that Shubhani Mukherjee has been doing. Dr. Mukherjee is also working on the Navigating Neuro HIV working group, has been doing fantastic work on that as well. But many of us know Shubhani's publication on neuroactive steroids that she published. And if you look on the left here, these are some of the neuroactive steroids that she measured, including DHEAS and pregnenolone. And she compared these particularly to depressive symptoms. And you can see that some of these effect sizes are quite large. Shubhani also compared these to indicators of immune activation, myeloid activation, inflammation. And you can see some strong effect sizes, again, including here for MCP1 and DHEAS and pregnenolone again. And so some nice preliminary data that supported a clinical trial that Dr. Mukherjee is enrolling in Boston called the SOOTHE trial, in which she's randomizing people with major depressive disorder to either pregnenolone 50 milligrams a day or placebo. And she is doing both neurobehavioral testing as well as brain imaging. And so this study is actively enrolling. So if you know of anyone up in Boston who would be interested in joining Dr. Mukherjee's trial, I think she might be grateful for that. And we're hopeful that once the trial is over, she might have some pretty impressive results. You're probably also aware of these data from the IAS meeting in Brisbane in 2023 on Ruxolitinib. And the Emory group was involved in these studies. Ruxolitinib, as you know, is an orally bioavailable JAK1/2 inhibitor. It's FDA approved for other conditions, but because of its anti-inflammatory, anti-JAK effects, they also looked at it with the hypothesis that it would reduce the HIV reservoir. And they had planned stratified analyses in people with HIV by baseline reservoir. And what they found was that in people with a high baseline reservoir, that there was a substantial decline over time. And that makes sense, that it would be, you'd see a decline most in people who had the highest HIV reservoir to begin with. And in their decay modeling, they predicted 99.99% clearance of the peripheral reservoir after about 3 years. So pretty impressive. And based on data like these and others, the Emory group has been moving forward with clinical trials of a next generation JAK inhibitor, Baricitinib. And they're performing 2 phase 2 clinical trials. Bert Anderson was involved with the first one here on reduction of HIV in the CNS, and I think in both of them. His mentor, Bill Tyre, has taken over this along with others, Vince Marconi and others at Emory. And Andy Miller, who many of us know, is leading this clinical trial for inflammation and depression. And both trials randomize people to either Baricitinib 2 milligrams a day or placebo for 10 weeks. And you can see some of the eligibility criteria here. These trials are only in Atlanta. So again, if you know anyone in the Atlanta area who would want to participate, I'm sure Andy and Bill would appreciate your referrals. Andy provided this nice graphic on the right here that summarizes the assessments and the depression trial. So this study is enrolling and going on. And again, I think it's potentially very, very promising, this Baricitinib treatment. Carl Goodkin, who many of us know, psychiatrist in the field for many years, working with the ACDG, anticipated this movement toward brain biotypes in trying to advance this clinical trial for many years. He very admirably dealt with a lot of issues, challenges, and questions within the ACDG to finally move this trial to enrollment. And what it's about is it focuses not just on people with major depressive disorder with HIV, but people with a combination of major depressive disorder and mild neurocognitive disorder. So sort of that neurobehavioral phenotype group 3 that I showed you earlier, the combination of depression and cognitive impairment. And the trial is based on Pramipexole. So as we know, commercial antidepressants that we typically use modulate serotonin, and some of them modulate norepinephrine as well. Pramipexole modulates dopamine, and the antidepressant effect was observed in clinical trials of people with Parkinson's disease. So David Moore and people from our staff are involved in the neurocognitive assessment and mood assessments in this trial, and they've been working hard to try to move this forward, and hopefully it will be enrolling very soon. One more clinical trial focused on the brain I thought I would mention has to do with CRISPR-based editing, and the work's being done by Trish Burdo and Jen Gordon and Kamel Khalili in Philadelphia at Temple, and the trial is enrolling in Philadelphia. But this busy slide summarizes some of their also very impressive data. And what the blot in the center of the slide shows is that the treatment results in HIV DNA being excised from nuclear DNA. So the DNA that they're looking at is a partial, a damaged DNA, part of DNA of HIV. Sorry. I meant to review the wording of that again and sort of stumbled on it, but you take my point. The CRISPR-based editing is removing part of the HIV DNA so that intact HIV RNA DNA is not being transcribed to RNA, and therefore proteins are not being made. So this is a nice way of potentially achieving at least functional cure. And so there's this excision trial. Trish was nice enough to share this slide with me. They've moved this from macaques into humans now with a lot of work. And this trial is ongoing. So if you know of anyone in the Philadelphia area who would like to participate, this is something that I think is also very promising. And again, as we all know, all of those researchers are very focused on the brain as well as HIV in general. So what about clinical trials with brain health as a secondary aim? So the first one I thought I'd mention is another one that involves another JAK-STAT inhibitor, Dasatinib. So this is being done by Alan Landay and Mary Claire Masters and Frank Palella from Northwestern in the ACTG, and it's focused on biological aging. This combination of dasatinib and quercetin are what people are calling senolytics. So they're meant to treat and hopefully reverse biological aging. So Mary Claire, Frank, and Alan and others worked hard also to get this through the ACTG, and it's a real triumph that they did. And they're going to not only focus on indicators of biological aging, but also physical function, so free frailty phenotype, gait speed, and so on. But they are also including assessments of cognitive performance and mood and sleep and so on. And that's, what in the ACG, that's what we try to get people, the point we try to get people to is, can we relatively inobtrusively include a few assessments, even if it's not the primary aim of your trial? Can we include a few assessments that we can learn something from your results? And so Mary Claire and Alan and Frank were very accommodating in that regard. And you can see here they're going to be looking at change in neurobehavioral performance, these senescence-associated secretory phenotype biomarkers, as well as these other indicators of physical functioning like time, chair, stand, and overall physical function. So I think this is going to be a very intriguing clinical trial as well. So Sarah Janella from UCSD and Peter Hunt from UCSF also struggled, devoted a lot of energy and attention to moving forward this clinical trial of Letermovir, CMV specific drug. And based on a lot of data they have, and I'm just showing a little bit of the data from our group, looking at anti-CMV IgG and showing, among other things, that it's associated with higher HIV RNA levels in CSF in people who are not on therapy, and worse cognitive performance in people who are on therapy. And so the plan for this clinical trial was to randomize 180 people to either 480 milligrams once daily or no Letermovir. So it's not placebo controlled, it's just no Letermovir. But the network required that they perform a futility analysis after they reached 40 participants reached their 8-week visit. And I won't go into the reasons for that, but I'll just say that they also required for that they, of course, identify one outcome that could be measured to support the futility analysis. And that was hard to do. And they ended up choosing soluble TNF receptor P75, soluble TNF receptor 2 concentrations in blood, because preliminary data supported that inhibiting CMV would reduce soluble TNF receptors in blood. And so they thought that would be the low-hanging fruit. But unfortunately, instead of going down, the soluble TNF receptor 2 in the Letermovir arm went up at 8 weeks. And so the Data Safety Monitoring Board recommended that they stop the trial for futility, which was a huge disappointment because they devoted so much effort to this trial. I learned so much from Peter, particularly about not giving up with what is, an apparent failure, right? And so I'm going to do quotes here for failure, but failure is the greatest teacher, right? And I'm making air quotes because this is not a failure, right? It's simply a clinical trial result, but it's a disappointing one. But Peter soldiered on because he's amazing. And what he found, among other things, what the group found with further work was they performed some O-link proteomics analyses, and they found that people who were randomized to Letermovir had reductions in IL-6 receptor, AXL, and Gas6. And these three proteins are ones that have been causally linked to cardiovascular disease. And so, and that was one of the other focuses of the work, not just brain health, of course, but cardiovascular or vascular disease. And so this was very supportive. And then they got a late breaker, was presented at CROI last year. They got a late breaker for this year's CROI based on newer findings on using the data that were generated prior to the trial stopping. Newer findings on the beneficial effects of Letermovir on physical functioning and other indicators. So, my opinion is that they were made to choose one biomarker here, and it ended up not being the right biomarker, and that decision had consequences. And there's a lot to discuss there. I don't want to get political, but I do want to just emphasize that Sarah and Peter have been doing fantastic work, and this is another area of treating, inhibiting herpes viruses, particularly CMV, that may be beneficial moving forward. Another area is long-acting therapies. And so this beautiful clinical trial called LATITUDE was performed in the ACTG as well. And as we know, many of our cities struggle with people who are addicted to drugs, and that really affects their ability to take oral antiretrovirals. And so we have people who are unable to maintain viral suppression, which is not only bad for their health, but also allows them to continue to transmit HIV as well. So that's one area that long-acting therapies are particularly promising in. And this trial focused on people like that, and randomized people to either getting cabotegravir, rilpivirine, long-acting every 4 weeks, or standard of care antiretroviral therapy, and followed them over time. And again, often in clinical trials, they will exclude people who are actively using drugs. This trial did not, because that was one of the focuses. And so it really helps us learn something about how something like suppressing HIV with long-acting therapy will affect not only HIV suppression, but also people's ongoing drug use, which obviously affects the brain too. So the results were really impressive. They were also presented at CROI in 2024, but you see here SOC is standard of care versus the long-acting formula. And this trial was also stopped early, not for futility, but for effect, that it was no longer safe to continue to randomize people to standard of care because they'd proven their hypothesis. The long-acting arm was so much more effective. So we've been trying to get funding for something like this to focus on methamphetamine users here in San Diego. This is a pretty graphic that Cecilia Marcondes made to summarize our program project application. But the project one here at the top was the foundation of the program, and that it would go into the clinic, work with folks at Owen Clinic here, and follow people as they started methamphetamine users, as they started ART, long-acting ART, and long-acting medication-assisted therapy, MAT, for methamphetamine. And we were going to do a number of assessments here in the trial, in the program, but we did not get funding for that. But this is a real growth area in the field. As you know, there's a lot of new technologies that are being developed to deliver long-acting drugs, not only antiretroviral therapy, but many other therapies as well, including, as I said, medication-assisted therapy for drug users. The final slide in this part is this one, which was provided by Katie Barr, and is just reminding us too that there are a lot of interventional trials for HIV cure. So Serena Sputic has worked in this area with the cure group in the ACTG. Marina Kasky and Katie Barr are the leaders of that group, but you can see summarized nicely here in the middle, the open clinical trials. There are multiple ones. Almost all include broadly neutralizing antibodies, or BNAbs. And Rebecca Wienhaus at Hopkins is waiting on review score for a very nice study that she's proposed, looking at distribution of broadly neutralizing antibodies into the central nervous system in macaques. So hopefully more coming in this area also. So finally, treatable traits. I learned about this from Bruce Brew as part of the Navigating Neuro-HIV Working Group. But this is a term that has come out of the pulmonary field that recognizes the complexity of lung disorders, because what they were seeing is that not everyone was responding to the same therapy in the clinic, and not everyone with lung disorders. And that sounds familiar to us with brain health disorders as well. And so this is getting adopted in other fields, this idea. But for me, it boils down a little bit to what we've said all along, is if we don't have treatments, specific therapies for brain health disorders in people with HIV, treat what you can. If they have diabetes, treat their diabetes. If they have hypertension, treat their hypertension. If they have lung disease, treat their lung disease, and so on. So, but as this area develops, they're defining key elements, that there is multidimensional, and the goal is that in the clinic, at least, and maybe in clinical trials, we'll have more personalized medicine. This graphic is similar to ones you've seen before, where, instead of on the left treating everyone as if they're just a homogeneous lump, we recognize the complexity of humanity and all the factors that influence health, and tailor the therapies to each person. So in that regard, treating people's lipids. So a lot of people with HIV are more likely to have dyslipidemia than people without HIV. And as we know, it puts them at risk for cardiovascular and cerebrovascular disease. And these data came out in 2023 from Steve Grinspoon, who also worked really hard to enroll this very large study. But Reprieve is randomized people to either get Pitavastatin or placebo. And you can see that the time to the first major adverse cardiovascular event, or MACE, differed substantially between the arms. I think this trial was also stopped early because it achieved its outcome. And you can see some of the, in this forest plot, some of the outcomes that they looked at with Pitavastatin versus placebo. So again, I won't dwell on this, but it falls into that, treating traits, treat dyslipidemia. You might remember from the Charter Aging study, we followed people over 12 years and looked at global cognitive change and the predictors of decline. And in this publication, which I won't dwell on a lot, but we found some of the factors that we expected to find, like diabetes and hypertension, and we had seen them before. But we also didn't expect to see lung disease. And so this chronic pulmonary disease was associated with cognitive decline over 12 years, whether people had it at their first visit 12 years earlier or had it at their second visit 12 years later. And so we're very interested in this idea of the lung-brain axis. And this was reinforced when we went to look at what therapies people were on. So we looked at some of these other medical disorders you can see listed here. We then looked within each of these groups of people with these disorders to see whether or not they were on therapy. They reported being on therapy for it or not. And you can see the proportion who did, roughly from a third to 3/4, which is not pretty far from universal treatment of these conditions. But what we saw when we looked at whether or not being on treatment made a difference, the only one that did was being on bronchodilators for chronic pulmonary disease. Those people who said they had chronic pulmonary disease and were on a bronchodilator did better over 12 years than people who had chronic pulmonary disease without a bronchodilator. We didn't see any effect for diabetes, hypertension, hyperlipidemia, but these analyses don't account for how effective the therapy for diabetes was, or hypertension, and so on. And we actually saw that people declined more if they were on treatment for depression or neuropathic pain. And Dave Grillotti checked me on this one time, appropriately so, and said, we don't want to give people the idea that being on antidepressants is bad for your cognition. And he's absolutely right. People who have more severe depression or a treatment-resistant depression are more likely to stay on antidepressants. People who have more severe pain are more likely to be put on stronger analgesics like opiates. So this approach does not, these analyses don't account for the severity of the disease. So just be mindful of the potential bias in the analysis. Leah focuses on women, and we all should because they're important. And another trial that Leah's been involved in as well, I think, in the ACDG is the HOP trial for hormone therapy. So you may know that for a long time we wanted to do no harm, so we didn't pursue offering menopausal, postmenopausal, perimenopausal hormone therapy to women with HIV because it could increase their risk for cancer, even though it reduced their risk for cardiovascular disease. But you know, the movement is to allow people to make their own decisions, provide data to people, information, and let them decide whether or not they'd like to participate or not. And so trials like this are finally moving forward. This one's randomizing people to either getting estradiol gel with or without progesterone, depending on whether they have a uterus or not, or placebo. And they're tracking vasomotor symptoms, but this trial also includes a neurocognitive battery, sleep questionnaires, mental health questionnaires, and so on. So again, this is a great way of integrating things that are important in brain health into a clinical trial that's being done anyway, so that we can learn something about how these interventions affect the brain. And I'll end this section just on GLP-1 receptor agonists. So these are drugs that you see listed here whose use is really increasing for diabetes, obesity. They also reduce cravings. In this ridiculously complex graph on the right, I love this graph. It's a Circo's graph. I hadn't seen something exactly like it before, but it summarizes a lot of information in one graph. And so what these authors did was they went through the literature and summarized all of the conditions that were reported for use of these GLP-1 receptor agonists. And I wanted to take you through this circle by circle, but we're almost out of time. So I will apologize, but refer you to the publication, but show you this forest plot, which shows some of the same data, and draw your attention to these conditions that they've categorized as mental and nervous system. And you can see that there are reports that have shown that there are benefits of GLP-1 receptor agonists on seizures, neurocognitive disorders. Of course, we're familiar with the data on alcohol use disorders. Nora has published on that, schizophrenia, and other substance use disorders. And the effects of GLP-1 receptor agonists are perhaps diverse, as summarized by the nice graphic on the right, including brain insulin resistance, which Norm Howie and others at Hopkins have focused on, and growth factors, which Stuart Lipton and many others have focused on in the past as well. A range of effects, potential beneficial effects on the brain, and something certainly worth pursuing in the future. So a few additional considerations, and then I'll stop talking. I haven't talked at all about imaging, or hardly at all about imaging. And so Bo Ansis and others here, Aristoteris, are applying clustering methods to findings from imaging as well, with great effort. And so that's something else that we absolutely should be thinking about, how to work into clinical trials, but it of course also adds cost. So that is challenging, but not insurmountable. We, in the Charter Aging study, remember this was, we assessed people 12 years apart. We measured a panel of proteins in that clinical trial. And as we were analyzing them, we saw this fascinating correlation of some of them very strongly. So in these graphs on the left, the dashed line is the line of identity. And you can see there's some variation between the first visit on the horizontal axis and the second visit on the 12 years later on the vertical axis. But the degree of agreement 12 years apart in CSF is really remarkable. And Bob Heaton said, well, that reminds me of sort of the trait versus state idea, that there are some conditions that are a trait that are maybe determined genetically. Ron Ellis had the same idea. And others that really vary day to day, hour to hour, week to week, month to month, year to year. And so, but you can see these correlation coefficients for p-tau, soluble TNF receptor 2 again, amyloid beta, total tau, really pretty high. And when we looked at what the correlates were of 12-year global change score with Mattia Trinfio and Asante Kampualala, it was this one that looks like a trait, soluble TNF receptors, and others that were more states, along with some other conditions. So, another thing to think about how we integrate this. And then finally, there's substantial regional variation. And these are just the charter sites. But, just in the characteristics I've listed here, there's a lot of variation from sites on the West Coast to sites in the middle of the country to sites on the East Coast. And in a multi-center clinical trial, we're lumping everyone together. But when it comes to brain health, people could be quite different in different regions. And that doesn't even mention, around the world. It doesn't even account for international sites. So I guess I lied. This is the final slide. So prescribed medications can affect brain health. This is something we recently published. As we're talking about putting people on more drugs as they get older, remember that polypharmacy is an issue in people with HIV. Just a larger number of drugs is associated with worse cognitive performance and more depressive symptoms. Anticholinergic burden, benzodiazepine use, antidepressant use, opiate use, these all can influence brain health. So many reasons could explain why we haven't been seeing positive results from clinical trials. Do we have the wrong target? Do we have, probably. Do we have the wrong outcome? Probably. Shifting demographic disease and treatment characteristics, regional differences, many others. A lot of people to thank for this. This is a terrible thing to show, but I want to encourage everyone to be thinking of people up in LA. We had a small fire a mile from my house here in San Diego also, but just remind us that even as these horrible fires blaze, we can't forget about them and have to attend to them. But peace of mind is still available to us if we turn toward it. We can't let it distract us from the work that needs to be done when these fires are raging, but keep calm and carry on. So as usual, I've left no time for questions, but I'm happy to stay as long as people want to stay.
That was absolutely fabulous, Scott.